In this lesson, we'll continue our discussion of muscle tissue by taking a look at something known as the sarcomere. We can think of this as the main functional or contractile unit of a muscle fiber. Then we'll take a closer look at the actual myofilaments. We introduced them in our last lesson, but we'll take a look at actin and myosin, what they're actually made of, and then we'll put it all together by looking at something known as the sliding filament theory. How do these myofilaments interact within the sarcomere to cause the muscle contraction? So let's start with the sarcomere. As I said before, the sarcomere, think of it as the smallest functional unit of a muscle fiber. To me, this is the part of the muscle fiber that causes the contraction. And this is basically happening because there's an interaction between the filaments, right? The myofilaments. We're going to see the thick filament and the thin filament interact to produce the contraction. Okay, um, so what we're going to look at now is the actual arrangement of the filaments to produce this sarcomere. We're going to see that there's two distinct areas. There's a dark area known as the A band, and then there's a light area known as the I band. So before I show you the parts of the A band and the parts of the I band, let's do this. Let's take a look at the actual sarcomere. So from this squiggly line right here, all the way over to the next squiggly line right here, everything in between is a sarcomere. So where did this come from? Let's go back to this picture. If you remember from my previous lesson, this whole structure here is one muscle cell, one muscle fiber, right? It's not a whole muscle, it's just one muscle fiber. Then these circular areas here were the myofibril, and then the red dots in it were the actual myofilaments, right? The actual actin and the myosin. They're in there. So what they did here is they pulled out one myofibril, and we can see the myofilaments kind of from far away. But if you remember from our previous lesson, the dark blue line was the myosin, and then the red lines that kind of overlapped were the actin. So what they're doing in this picture here is they're taking this and they're blowing it up, right? You can see the squiggly line right here. That represents that squiggly line right here. So when you look at this picture now, if you, from this line down here, if we kind of drew an imaginary line right there to here, this whole area is darker. So this is going to be what's the A band. But right next to it from here over to where it gets dark again, everything in here is going to be the I band. Okay, so let's take a look at the parts of the A band first. I'm just going to go back a slide just to follow in order. The first thing I want you to be able to identify and understand is known as the M line. Think of M as middle. So this line right here represents the M line, and what this is actually a bunch of proteins, and these proteins connect this thick filament to that thick filament to that thick filament to that thick filament, and it forms this line right down the middle. The function of this M line is to help hold these thick filaments in place. You're going to see over the next couple lessons we cannot have the thick filaments move. They need to stay stationary. Right? So that M line is actually proteins, right? It stays stabilized the position of the thick filament. Now, the next part within the A band is another smaller band known as the H band. What's happening here is we only see thick filaments but no thin filaments. So if you take a look just adjacent to the M line, there's thick filaments here only. Right? And on this side also, there's only thick filaments. We don't see any thin filaments. So that little bit of a lighter area, because there's only thick filaments, is known as the H band. Right? And again, that's a band within the bigger band, the A band. Before I go back to the notes, so just adjacent to the H band on this side, and just adjacent to the H band on this side, we see that there are both thick and thin filaments. They overlap one another. So this whole area that my pointer is going up and down on this side, and same thing over here, we call the zone of overlap. The zone of overlap, and that's just because the actin is overlapping the myosin. Okay, so the three parts of the A band I'd like you to know are the M line and their function, right? The function is to stabilize the position of the thick filaments. That's your M line. Then adjacent to it on both sides, there's only thick filaments, no thin filaments. That's going to be the H band. And then if you go over, again on both sides, zone of overlap. Okay. So that pretty much covers everything here, right? So the zone of overlap is going to look darker for sure because you have both thick and thin filaments. Okay, let's take a look at the I band now. Now the I band you're going to see contains thin filaments only and no thick filaments. Let's start here. So from the edge of the A band here, if we go all the way over to the end of this sarcomere, but watch this, we actually go into the adjacent sarcomere here, right? This is the next sarcomere to the left of the sarcomere that we're looking at. So the I band is partially in one sarcomere and the other part is in another sarcomere. What we see kind of bisecting the middle of the I band is known as the Z line. Right, so this zigzag line right here is the I band, and this is kind of what I use as the boundaries of one sarcomere. If you remember before, I said the sarcomere went from this Z line all the way over to this Z line, right? That's one compartment, and then this is the beginning of another compartment over here. All these compartments are attached to one another. Okay, so this is known as the Z line, and it bisects, right? Which just means that it cuts through the I band. And you can use it to mark the boundaries of adjacent sarcomeres. Again, it's what you want to look for to find one sarcomere and then the sarcomere next to it. Now, we said that the I band contains thin filaments, right? We're going to have the red lines in there. There is another filament in there. It's a protein. It's called Titan. Titan means that it was big, right? Like Titanic. When they saw it, they saw it was a big protein, and it's an elastic protein, which is going to be important in function. So I'm going to come back to these notes in a second. Let's go to the picture. So if you take a look here, guys, from the Z line, we have attached to it this squiggly green line going right to the side of the thick filament. Each one of these green lines is the Titan. Okay, the red lines attach from the Z line and they go over into the zone of overlap. So the green line is the Titan, the red line is the actin. All of these green lines here, the Titan, again, are going to attach into the side of the thick filament. This also helps to stabilize the position of the thick filament, because look at, we have it on this side also. Right, this Titan right here is attaching to the side of this thick filament. So between this Titan here, and if I go to the on the opposite side right there, that Titan there, that's holding this thick filament in place. It kind of reminds me of a hammock, right? If the Z line was a tree, right? Here's the rope, here's the hammock, and then here's the other rope going to the other tree, let's say. Okay, so it's kind of holding the thick filament in place. And so that's an important function, right? So it's an elastic protein. It extends from the tips of the thick filament over to the Z line, right? So each squiggly line, if you looked at it there, the green one would go from the Z line to the thick filament. And like I said, it keeps these filaments, that if you want to even add here, the thick filaments in proper alignment, right? So its function is very similar to the M line, but it also helps, since it's elastic, restoring the resting sarcomere length after the contraction is done. It actually helps to bring the sarcomere back to its original length. That's because it's an elastic protein, right? It provides a little bit of recoil. Okay, so all of this is the I band. So let me just go back to our very first lesson on muscle tissue. I had said to you that muscle tissue is striated, and striations were those patterns of light and dark. So now when you look in the microscope and you see light, right next to it, dark, right next to it, light, dark, light, dark, light, dark. The C line is actually dark, light, and it's these patterns, the arrangement of the actin and the myosin and the titin, all this that gives us that striated appearance when we look at a muscle fiber underneath the microscope. You know, areas of light, dark, and light. You can even see up here. Here's a muscle fiber, light, dark, light, dark over here, that type of thing. I love this picture here. This is an actual micrograph of the sarcomere. Look at this. We magnified this 64,000 times. Our microscopes in the lab that we use only magnify up to 1,000 times. So this is really, really magnified. But just to kind of give you an idea, what we were just looking at, if we go back to following our notes, I'll follow the same pattern. Here's the A band. That's going to be the dark. So from here where there's thick filaments, all the way over to the thick filaments over here. That's going to be the A band. Then this would be the M line in the middle. And then in the light area just to the side of it, H band. Right? And then there's an H band over here. And then this is going to be actin and myosin. This is going to be the zone of overlap on this side, zone of overlap on this side. In the I band, here's your Z line. Now we don't see the actual individual actin and the titin, but those guys are all over here, right? The actin is all projecting towards the middle to the zone of overlap, and again, the titin is just going to the side of the myosin. All right, so that's an actual micrograph of the sarcomere. This is more of an FYI, just if you're interested in studying this, you can. This is just giving you a perspective where they actually slice through the muscle fiber at various places within the sarcomere to show you the fibers. So if we were to slice here, this is actually through the Z line. We can see the thin filaments are the red dots, all these red dots right here. Those are the thin filaments. I didn't mention this before. Actinin is another protein that actually ties the thin filaments on one side of a sarcomere with the thin filaments on the other side of the sarcomere. It's known as actinin. So it's a protein that's actually in the Z line connecting, let's say, this thin filament to that thin filament. So we can see these little lines right here are the actinin. Then if we cut in the middle of the I band here, we would see the red dots, which are the thin filament, and then the green is going to be the titin. Then if we cut through the M line, right, these are the different proteins now that are connecting the thick filament, right? So there's going to be the thick filament and the proteins connecting them. Then over here, this one here, this is going to be where the H band is right here. And there we see the thick filaments only. And then over here, they're giving you the zone of overlap. They didn't do the zone of overlap over here, but we're doing the zone of overlap on this side. You can see both the thick and the thin filaments. So it's just giving you a perspective on cross-section rather than looking at it head-on, from the side, I should say. Okay. This is an interesting slide too. Here they're actually taking you through from an entire skeletal muscle down to the sarcomere. This is called the levels of functional organization. This is going to kind of take everything that we've learned so far and kind of put it together. The biggest level of organization is the skeletal muscle. So just to kind of review, we saw the skeletal muscle had the epimysium around it, right? And then here you can see the bundles within the skeletal muscle. These were called the fascicles. So here, guys, they're going to pull out one fascicle, and here they're blowing up a fascicle. And we see that the fascicle had the perimysium. And then within the fascicle, there are between, we said in the previous lesson, 10 and 100 muscle fibers, right? Muscle cells. Here's a muscle cell. Okay, so now they pulled the muscle cell out and we're looking at it, right? We can call a muscle cell a muscle fiber. And we see here the connective tissue around the muscle fiber or the muscle cell was the endomysium. Then within there, the red dots, these were the myofibrils, right? This is where the actin and the myosin is. So here they're pulling out the myofibril, and we're looking at that over here. Right, here's the myofibril, so we can see the myofibril had like the thin and the thick filaments. Here's the SR. We saw the T tubule. Right, this whole thing here was the triad. Then they take the myofibril and they just take out one compartment between Z line to Z line right over here, and this is going to be our sarcomere, right, which we just studied. And you can see the A band, right, the H band, the M line, zone of overlap, I band over here. Right, on this side also, and then the two Z lines. So it just kind of takes us from an entire skeletal muscle and breaks it down into smaller and smaller functional units, right, functional parts, until we finally get to the sarcomere, which is really doing all the work. Okay, so now that we know the anatomy of the sarcomere, let's take a closer look at these thin and thick filaments. I've mentioned them, numerous times over the last couple lessons. Let's take a look at what they're made of. So we'll start with the thin filament. In actuality, the thin filament is made up of four different proteins we're going to look at. There's something known as F-actin, where F stands for filamentous. Right, it's kind of forming a fiber like a filament. Then we're going to see something called nebulin, and then what I call two regulatory proteins, tropomyosin and troponin. Right? Some people say troponin. So before I go through these notes, let's take a picture here. Take a look at a picture actually. So what the book did here, guys, watch this. If you recognize the sarcomere, here's the Z line right here. And extending from the Z line, this little red line is an actin strand, right, and myofilament actin. So what they did is they blew it up. So we can see here's the actin, right, that would be the one on the left over here. Then here's the one on the right going this way. So now we've taken this actin and we're going to blow it up this way, okay? And I blew it up even a little bit more. So now we have the actin. So what we see is this. This whole strand of actin is known as the F actin strand. It's the filament, it's the fiber, right? That's what the F stands for. But this F actin strand is actually made up of these small globular proteins, right? There's about 300 to 400 of these globular proteins called G actin. Globular means that the protein is kind of round and circular. So we have all these G actins. They are tied together to form this strand known as the F actin. What holds these little G actins tied together right here, see this blue line right down the middle? That blue line kind of reminds me of like a rope that's running right down the middle, and all the round balls there are attached to it. That rope, right, that protein that runs right down the middle, that blue one, that is known as nebulin. And again, the nebulin is tying all these G actins together so that we form this entire strand known as the F actin. So let's go back real quick. All right, so the filamentous actin, or you could just say F-actin, is a twisted strand. Right? That makes the filament composed of two rows of these globular, which just means round, G-actin molecules. Now, when we look at these G-actin molecules a little bit closer, you're going to see there's like on this diagram, there's a little yellow area. Right, so it's kind of orange and then yellow. That yellow area is what we call an active site. That's going to become important in our next lesson. The active site is going to be the place where the myosin, which is the thick filament, actually connects to the actin. So these active sites are important. On this picture here, you can see the active sites are covered by this kind of turquoise looking braided structure here. That's one of the regulatory proteins I'm about to describe now. Okay, so what do we need to know so far? The entire strand you're going to call F-actin, filamentous actin. What is it made of? It's made up of these about 3 to 400 of these G-actin molecules. There's two rows of them. right? They're all tied together by this blue line right there, known as nebulin, right? So that nebulin is running through the entire strand, and all those G-actins are tied to it. And then it's important to know that the G-actin is going to have an area known as the active site. That on this picture here, it's all these yellow top areas right here. And then again, in the next lesson, we'll see that the active sites are important because they are going to allow for the myosin to attach to the actin. Just jumping ahead a little bit, we'll see in order to get muscle contraction, the myosin has to attach to the actin. Okay. All right, so we got that, and then we said the nebulin holds the actin, the F-actin strand together, right, so that it forms a strand. So let's talk about these regulatory proteins. There are two regulatory proteins. Basically, they're the proteins that are going to allow muscle contraction to occur or not. The first one is known as tropomyosin. This is kind of like a braided strand that covers the active sites. Let's go back to the big picture. So this braid right here, this is a protein. If you notice, guys, it's covering all of the yellow areas right here. So if this braided strand is covering all of the yellow areas, that means the active sites cannot interact with myosin. And this muscle fiber then would be in a relaxed state. There's no contraction taking place. All right, so that braided strand right there is tropomyosin. Now, that protein right there, right, we can see it here. Notice this protein is attached to the tropomyosin. It's also attached to the G-actin. This protein here is called troponin, or some people say troponin. What happens is this. Troponin, when it receives an ion, which I'm going to talk about in the next lesson, known as calcium, calcium is going to bind to the troponin. The troponin is going to change its shape and pull the tropomyosin out of the way, exposing the active sites. We actually see that a lot in anatomy where a protein changes its shape to do some type of function. So when the calcium, right, which is released from the sarcoplasmic reticulum. If you remember from a previous lesson, the sarcoplasmic reticulum, its job is to store calcium. When the calcium is released, it's going to bind to the troponin. The troponin is going to change its shape. That's going to pull this braided strand, the tropomyosin, out of the way, and now the active site will become exposed. Once that active site is exposed, muscle contraction will take place. So let me go back a minute. We'll take a look at the notes. All right, so tropomyosin, I want you to know, covers the active site and it prevents the actin and myosin from interacting. If you just want to add, this would be a muscle at rest, a muscle fiber, not a muscle, put a muscle fiber at rest. Right, so that's what's happening. That tropomyosin is covering the active site. The troponin, right, or the troponin, however you want to pronounce it, this is a globular protein, right? It's actually attached to the tropomyosin. It's attached to the G-actin, and it has the site for this stuff here, the calcium. So if you just want to add a note, when the calcium binds to the troponin, it changes its shape, pulling the tropomyosin out of the way. Right? I'll repeat that. When the calcium binds to the troponin changes its shape, pulling the tropomyosin away from the active site. Kind of something like that. That's the concept. So I like to call these regulatory proteins because they're regulating muscle contraction. Okay? So this is a great picture. Again, what do you need to know? The whole strand is the F-actin. It is composed of two rows of these balls here, right? They look like balls, like ping pong balls. They are called G-actin, which stands for globular. That means they're round. These balls are attached together or held together. These proteins, I should say, are held together by this string right here in the middle, or this rope right in the middle. It runs all the way through. We see it here. We see it here again, there. We lose it, but it's there, and then it's back again. That whole strand is called nebulin. Then the active sites on the G proteins, they are covered when a muscle is at rest by this braided protein here called tropomyosin. And that tropomyosin is connected to a regulatory protein known as troponin. And when this troponin receives calcium, it changes its shape, pulling the tropomyosin off of the active sites so that muscle contraction can take place. Okay, so that's the thin filament. Let's take a look at the thick filament now. Remember we said there was two filaments that form the myofilaments, the thin and the thick. So the thick filament is made up of myosin molecules. Right? It's about 300 myosin molecules combined together to make a thick filament. So before I go over the notes, let's go to the picture. So what they did here was they took a thick filament, just to show you where we're at. Here was the M line. Remember, the M line was the proteins that connected the thick filaments together so they don't move. So here's a picture of the M line. And then showing also the titin, that was that green squiggly line coming to the side of the thick filament. So now guys, if we look at a thick filament here, what we're going to see, there's a combination of about 300 individual myosin molecules like bunched together, right? They're going to be grouped together. The anatomy of the myosin molecule is such, it kind of reminds me of a golf club, right? If you have this long handle right here, this is called the myosin tail. At the end of the golf club, we have a head, right? That's what would strike the ball. However, unlike a golf club, this head has the ability to pivot. The reason for that is there's a little hinge right here. That's going to become important in our next lesson when I show you how a muscle fiber actually contracts. So just kind of know that the the myosin head has the ability to pivot, or swivel is the word we actually use to describe that. Okay, so the tail, that's going to be connected to all of the myosin molecules, right? You'll see all the tails are kind of connected together, and then projecting off, these are the heads, and the heads would be projecting towards the thin filament. If you follow where my cursor is right here, where the thin filament would be right here. So these myosin heads, when the active site is exposed, will actually attach to the thin filament. Okay, so these myosin heads project off towards the thin filament. And then again, remember we said the titin, we talked about this already, but just to kind of show you, this titin here helps to hold the thick filament in place, but also, since it's an elastic protein, it helps to reestablish the length of the sarcomere after contraction. Okay, so that's what our thick filament looks like. So let's see if we could put this all together, right? We know the sarcomere. We know the structure of the thick and the thin filament. What happens during a muscle contraction is this, a muscle fiber contracting, I should say. It's something known as the sliding filament theory. Basically, what's going to happen, let me go back to this picture here just to kind of give you an overview. Nice big one. Let's go to this picture. Let's go with this picture here. When a muscle fiber contracts, guys, this is what's going to happen. The myosin heads on the thick filament are going to attach to the thin filament. And then what they do is they pivot, they swivel, and they pull the actin towards the middle of the sarcomere. And what that does is that pulls this Z line in, and then this Z line in this way, and the whole sarcomere shortens. So basically, with the sliding filament theory, the actin is going to be sliding over the myosin. So when that happens, we see that the H band and the I band are going to narrow. So check this out. Here's your thick filament. Right? These little dots sticking up here, these points sticking up, are the myosin heads. Remember we said they point towards the actin. So here is a muscle fiber when it's relaxed, right? It's kind of in the relaxed state right here. We see the relaxed state, this picture. So we have our H band that has a certain width, right? The I band has a certain width. Now, when the muscle fiber is going to contract, the myosin heads, they jump up and they attach to the actin, and then they swivel this way. And we're going to see that the actin is going to get pulled towards the center of the sarcomere on this side, and this actin here would get pulled towards the center of the sarcomere on this side. Right? So all of these thin filaments get pulled this way, and then all these thin filaments get pulled this way. So watch what happens when we go to the contracted state. Notice now the muscle fiber is contracted. See how the I band here has gotten smaller. The H band has also gotten smaller. The zone of overlap has increased, and that's because the actin was pulled across the myosin. Let's go back. See the zone of overlap here? Right? Maybe goes 1/3 of the way over, but look at now when the actin gets pulled over, right? It's almost 3/4 of the sarcomere or the A band that we have overlap, right? So we increase the amount of overlap from this amount to this amount, right? And then again, go back, look at the I band, how big it is here. This part of the I band, when we go to the contraction, see how it got smaller, and the Z lines got pulled closer together, right? The two Z lines got pulled closer together, and this sarcomere here is shortening at the same time. This sarcomere is shortening, this sarcomere is shortening. They're all shortening, so the whole muscle fiber, right, the myofibril, is going to shorten. Like I said, kind of like a slinky. Okay, so during a muscle contraction, right, we're going to see the H bands and the I bands are going to get smaller. The zone of overlap is going to get bigger, right? We're going to see more overlap, and the two Z lines are going to come together. The whole width of the A band actually stays the same. The A band, right, the thick filament to this thick filament here, that does not change, right? Because that's stationary. Only the thin filaments are sliding over the thick filaments. And again, you can see here, if we take all the sarcomeres together and they contract, the overall effect is the whole muscle fiber is going to, or in this case, the myofibril, but when you have all the myofibrils contracting together, the whole muscle fiber is going to shorten. So this is what we call the sliding filament theory. Right? There are a couple of nice videos to accompany this so that you can watch it and see it more visually. And that's it. That's how our muscle fibers contract.